Welcome back. Hope you had a nice break. I'm Terri Combs-Orme, presenting this course for you for continuing education credit. Thanks to the funding and support of the College of Social Work of the University of Tennessee and the Urban Child Institute. There are no restrictions on use of this material, and if you have questions or just want more information, email me at the address you see. So we concluded the last set of slides by talking about the Human Genome Project, and I emphasized that we did not learn the origins of all traits and diseases. We didn't even come close. In fact, probably one of the most important things, or some of the most important things we learned from the Human Genome Project was what we did not learn. Social work has always talked about the interaction between genes and environment, and it turns out we were right. But the truth is, it's way more complicated than that. We have so many alleles for so many traits and disorders. Some are expressed and some are never expressed. So how does that happen? We know the environment has the influence, but how does it do that? Remember again, we talked about those 4 proteins that make up our chromosomes and how they combine in all different sequences to code for for different traits and characteristics. It is actually through the production of proteins that our DNA codes work. And genes don't automatically express their proteins. Something else spurs them to be expressed or not. If a gene can't produce a protein, we say it's not expressed, so that it's not part of your phenotype. So we're going to talk about what kinds of things cause those genes to be expressed. And I think it's really exciting information for social work practice. So gene expression happens in one of 3 ways that we understand as of the day I'm doing this tape. We're not going to talk a whole lot about the last one, but which are non-coding RNA strands. But I will say that some of these processes come from that junk DNA that we called junk in the beginning. So it turns out it's not so much junk. Each of these methods changes expression of the genes in unique biochemical ways. So let's talk about the first one, methylation. This is the one we've done the most research on and that we understand the best. And it's really quite simple. Methylation is just a chemical modification of part of the DNA that allows a gene to be expressed or turns it off. And specifically, methyl group is added to a cytosine molecule. Remember, that's one of those 4 proteins. This can happen due to your diet, due to a chemical in the environment. It happens naturally with aging. One example, for instance, are tumor suppressor genes. We used to think that cancers were all caused by genetic mutations, but as it turns out, sometimes cancer is just caused by an epigenetic influence that turns off or partly disables the expression of tumor suppressor genes. Those are genes that are very, very active in stopping the overgrowth of cells in cancer that happens all the time in all of us. So when those genes get silenced or their activity is lowered, cancer has more of a chance to spread. And this is not a mutation. It's a simple modification of how the gene is expressed.
We will talk much more about this later, but changes in gene expression accumulate over life due to different experiences and exposures. As those changes accumulate, the genome is made more unstable, and cells and then organs are damaged.
You see a picture here, and the little red dots represent methyl groups that have been added to cytosines on this strand of DNA. Now, the other biochemical way that we understand very well is chromatin remodeling. Chromatin is just that complex of DNA and histone proteins that the DNA wraps around. They're just little spheres that help the double helix stay wound. It can happen, though, that the DNA can begin to unwind. So when that happens, some genes will be more accessible, so they will be expressed. Other genes, if the chromatin tightens up, genes can be silenced because there's not access to them anymore. So here you see a representation of that. When something causes the chromatin to be bound up more tightly, genes can be hidden from expression. So why is this important? We understand now that it's not just what genes you have. It's what genes get turned on and off by the environment. And this is the subject of epigenetics. Hold on, though. This is great news for social work. It's not the genes you have. It's the genes that are expressed. And you're going to see in a moment why it's not the genes you have. It's the genes that are expressed. Because chromatin remodeling and methylation can happen throughout life. Just for emphasis. Let me give you a few examples of some of these processes that we understand. We understand some a lot better than others. Fetal alcohol syndrome, caused by excessive drinking during pregnancy, is caused in part by the methyl in alcohol, and it interrupts fetal cell methylation during key points of development of the fetus. So some genes that ought to be expressed aren't. So, for instance, many fetal alcohol syndrome children have heart defects because the methylation was disturbed when the heart was forming. They have different facial characteristics because methylation is disrupted during formation of the skeletal structure. All of these things explain the biochemical ways that alcohol has its detrimental effects during pregnancy. Other types of childhood, like leukemias, are caused by abnormal proteins in cells.
Acetyl groups may be added to histones, causing some cells to overgrow. Acetyl groups are simply methyl groups with added carbons.
And so the cell is not able to methylate and model in the way that it's supposed to. Interesting, methylation explains the use of prenatal vitamins. Prenatal vitamins are very high in folic acid, which is the synthetic version of folate. Folate, which is found naturally in green vegetables and other sort of healthy vegetable foods, is key to methylation during pregnancy. And because the standard American diet does not include generally enough folate to promote effective methylation,
many children were being born with spina bifida, a birth defect where the spine does not close up properly during development and remains exposed.
Prenatal vitamins that include folic acid replace some of that. And folic acid was added to bread and other baked products because the most important time for methylation, when folate is needed, is a time when most women do not even know they're pregnant. It happens around the time of conception, and taking them later can be helpful, but it's most important early when the brain itself is developing.
Now, as I mentioned earlier, changes in gene expression begin at birth.
And it ends at death. So the important thing to remember is that gene expression can be modified throughout life. Changes that occur due to various influences of the environment can often be reversed. And this is why it's big news for social work. So how do you do that? Let me give you a few examples. Diet changes gene expression. You often read or have heard on television that broccoli helps prevent cancer. Broccoli does this because compounds in the vegetable promote healthy methylation of the cells and particularly strengthen methylation and proper expression of the tumor suppressor genes that you have. Broccoli is truly a wonder food. Exercise changes gene expression, and it does this generally by affecting the genes that affect your metabolism. So when we talk about exercise preventing heart disease, and we talk about it preventing or being good for treatment of diabetes, it's because it encourages those genes that perform those functions to methylate appropriately. Many of the pesticides that we now know are poisonous for human consumption operate by changing gene expression. So they do not cause necessarily mutations to the genes, but they alter gene expression in very harmful ways that are seen in people's phenotypes. Most important for us as social workers, though, is the revelation that social conditions influence gene expression. Stress, that we'll talk about in the third lesson or the third class of this series, is directly related to gene expression because stress is a physiological process. Racism and discrimination, poverty, child abuse and neglect, and even non-nurturing parenting can influence gene expression. And this is huge information for us as social workers. It's even true that gene expression changes can be passed on to subsequent generations without affecting the DNA code itself. Now, the first scientist who suggested that experiences could be passed along to your progeny was laughed out of the room and humiliated and ended his career in a state of real humiliation. But it turns out that he was on to something. This does happen sometimes. DNA is not altered, but gene expression is changed. Now, one of the things that happens, because mothers and babies share a blood supply, everything the mother experiences is experienced by the baby. Famine, war, domestic violence, depression. The mother's physiological system is also the baby's system. So the baby shares all of this. So, for instance, in famine, a woman doesn't get enough to eat. It affects the developing fetus by changing some gene expression. War, may make a woman anxious and highly stressed and very reactive, trying to protect herself, and those impacts will be passed along to her fetus. Let's look at some important examples. The first example is called the Dutch Hunger Winter. In 1944, going into 1945, the Nazis blockaded the Netherlands and prevented food entering the country as punishment for some of the resistors who were struggling against the Nazis. Calories were cut hugely to about 800 calories per person. And pregnant women, need way more calories than that. So there was basically starvation throughout the country. The mortality rate in Amsterdam, for example, doubled in 1945. Babies born during this time were very much smaller than they normally are. And that's not terribly surprising because mother doesn't have enough nutrition if she's not eating enough. But it had more effects that were long-lasting. During the Dutch Hunger Winter, there were long-term effects on the fetuses who were gestating during that time that varied by sex and by gestational period of exposure. For example, men who are exposed to famine in early gestation are much more likely to be obese now. They have twice the rate of schizophrenia and empty personality disorders. Both sexes experience more depression and affective psychoses due to the famine. These individuals are about 70 years old now, and they have many chronic conditions. Fortunately, the Dutch keep very good records. And what we find is that these individuals are suffering much more cognitive decline with age than the general population. There is more breast cancer, more kidney disease, lots of heart disease. There's diabetes, obstructive airway disease, and all kinds of cancers. And this can all be traced back to the improper nutrition, malnutrition during gestation. More recently, a lot of other things have come to light. So it was once believed that the children and grandchildren of Holocaust survivors have high rates of PTSD and anxiety and depression because of the stories they heard from their families, from the survivors. We now know that those traumatic events were probably passed along in epigenetic changes to those individuals that make them more susceptible to stressful events that would not be disastrous for other people. But that gene expression change makes it very difficult for them. So, again, the interesting thing and the fun thing to read about is that several types of interventions can reduce or reverse these gene expression changes. Exercise has been shown to, for instance, increase functioning of the metabolism through stimulation of genes that do a better job of producing insulin and making the body sensitive to insulin. These changes actually show up in individuals' metabolisms. Diet has big changes. Some interventions have shown that the elderly, when they get adequate levels of folate, have much better cognitive functioning. And it's because of changes in various genes due to the better methylation. Finally, cross-fostering research has shown some serious and important effects of parenting and its effect on individuals. The key point for this entire lesson, and one I want to emphasize again, is that epigenetic changes can be reversed. Social workers have, in the past, been sort of hostile to the idea of the importance of genetics because it seemed to determine it. It seemed to suggest that you're stuck with what you get. We now know that different environmental conditions can change gene expression, so this is very exciting news for us. And we'll cover more of that in future lessons. Thank you.